Hello, my name is Carolina Brooke. I'll be guiding you through the remainder of the microbiology chapter. We'll start with virology, which tends to be a harder section to memorize. We'll start off by talking about the basic properties of a virus before going into specific viruses and what you need to know about them. First, we are going to discuss basic virus structure. You may be wondering, why does this matter to you? This is a really boring topic and you probably don't feel like studying it. However, on the step 1, in some questions, they will describe a virus to you based mostly on its structure, and then they will ask you to identify the virus. To know all of this, however, requires a lot of time and memorization and, to be honest, is not very high yield. So what I'm going to do in the rest of this lecture is try to point out viral structures that are important. And in fact, this should help you remember and understand much better their pathogenesis. So before we start going into the general features of a virus, I'm going to ask you a seemingly innocent question. What is a virus? You may think this is obvious, and probably from studying high school biology you might know the answer, but let's go over this together. Simply, viruses are obligate intracellular parasites that can only replicate using the cellular machinery of a host cell. All viruses are made of genetic material, known as nucleic acid. And this nucleic acid can be RNA or DNA. Another thing that can differentiate virus is the structure of the viral genome. In fact, it can have many different shapes. Some of the many shapes that a viral genome can take on are listed here. They can be single or double-stranded, they could be segmented or non-segmented, and they could be linear or circular. In these 3 images, you can see circular single-stranded RNA, single-stranded DNA, and double-stranded DNA genomes. Another thing to consider about the viral structure is whether the nucleic acid is encased within a protein coat called a capsid. In this first image of a naked virus, the outer capsid is not surrounded by anything else. Here I'm pointing to the outer capsid. Now note that the capsid itself can take on one of 2 shapes. It can take on an icosahedral shape, which you can see in this image, or a helical shape as shown in this image. So what are the purposes of the capsid? Well, the capsid acts as a protective shell that holds the viral genome and virus-encoded enzymes. The capsid is actually composed of proteins that contain receptor sites necessary for initiating infection by the naked viruses. Another function that the capsid has is that it also provides the antigenic stimulus for antibody production. As you can see in this third and final image on the right, there is another layer around the capsid. I'm pointing here to the capsid. Here is this extra layer. What does this do? This makes it an enveloped virus. Now, why is the envelope important? Well, the answer to that question is that part of the virus's infectivity is related to its envelope. An envelope can be inactivated by ether, which is a lipid solvent that causes the virus to lose its infectivity. In contrast, a naked virus is resistant to ether. As you can see here, the envelope is a lipoprotein bilayered membrane. The viral envelope is composed of lipoprotein membrane that is acquired from the host's plasma membrane during the virus's release from the host. The envelope contains virus-encoded glycoproteins which give the virus its ability to attach to host cells and initiate infection. The glycoproteins also stimulate antibody production by acting as antigens. So now we're going to talk about the genetics of viruses. Viruses are able to undergo processes that in some cases may change the viral genomes. Recombination and reassortment are 2 processes resulting in alterations to viral genomes that can lead to new strains. First, let's talk about recombination. Recombination involves the exchange of genetic material between 2 related viruses that have at least some similarity in their genes. This occurs during co-infection of a host cell. It involves the exchange of genetic material between homologous genes of 2 chromosomes. This image does a really great job of showing the idea of recombination. you can see very clearly how homologous genes of two chromosomes cross over, creating these recombinants. Reassortment is another process that can happen in the viral genome. Here's a case where it is actually useful to know the viral genome structure, and I'll explain why in a second. Reassortment is something that occurs specifically in viruses with segmented genomes. Influenza is an example of a virus that has a segmented genome and it is able to undergo reassortment. Why do you think this is important? Well, actually, this very basic science information is relevant to why influenza is so pathogenic, and I'll explain about that in a moment. So let's go through the process of reassortment. Imagine that a cell is infected with two different influenza viruses, each with 8 RNA segments. The RNAs of both viruses will be copied in the nucleus. When new virus particles are assembled at the plasma membrane, each of the 8 RNA segments may originate from either infecting virus. The progeny that inherit RNAs from both parents are called reassortants. The resulting change from reassortment is called antigenic shift. In influenza, this can contribute to worldwide influenza pandemics. Do you understand why this is the case? Well, if you go back and remember immunology, your body will form an immune response against antigens it has encountered before. However, when influenza mutates in this manner, creating these new viral strains, your body will not have immunity against this new strain. The next two processes I will talk about in viral genetics are actually phenotypic changes that do not change the viral genome. They include processes such as complementation and phenotypic mixing. Let's start with complementation. Complementation occurs when one virus rescues a defective mutant strain of a virus that is missing a gene for an enzyme or factor necessary for replication. In simple terms, just think of complementation as one virus helping the other. I scratch your back, you scratch my back, is the basic idea. Alright, let's take a look at this image. Look at this orange-colored virus. I've illustrated that it has a broken gene. It is drawn here with a dashed red line. Imagine that this red segment has a gene for a protein that is actually necessary for replication. However, if you take a look at this purple virus, you'll notice that that same red gene is drawn correctly. It does not have a dashed line. In fact, this red gene has that important factor intact. So what can happen is that these two viruses can co-replicate together. Our purple virus, the rescuer virus, will now be able to supply the missing factor or protein to our orange-colored defective virus. So, basically, the defective virus will now be able to replicate and form progeny. So let me ask you, what do you think the progeny will look like? Think about this for a second. Well, actually, the progeny of this mutant virus will also lack the same gene and won't be able to replicate unless they too are rescued. Do you understand why this is the case? Well, remember that this is only a phenotypic change. It will not change the genome of this virus, and that is why the progeny will also have the same mutation in that gene segment. Alright, let's move on to the last form of viral genetic process, phenotypic mixing. In phenotypic mixing, a single host cell is infected with two related viral strains. The genome of one strain, virus A, is packaged within the capsid of another, virus B, so that the outside capsid is composed of proteins encoded completely by virus B, but the inside of the virus contains genetic material from virus A. Now that sounds like a mouthful, so let me show you the images that correspond to this idea. So here we have virus A and here we have virus B. Both virus A and B are infecting the same host cell. This is the progeny that results from this co-infection. This is the process of phenotypic mixing. So what can you make of the progeny of phenotypic mixing? Note here that virus A has a green genome, while virus B has a yellow genome. Notice the colors of the outside envelopes for these two viruses. You'll notice, for example, in this virus, the outside envelope is orange colored, like in virus A, but the genome is yellow, like that of virus B. The same is true for this little virus that I'm pointing to here. The genome is the color of virus A, but the outside envelope is the color of virus B. In other words, we have complete mixing of the genomes and envelopes of the two viruses. In other words, you can think of the progeny of the pseudovirion will contain the same genetic material and capsid proteins as that encoded by virus A. However, remember that virus B capsid determines infectivity pattern of the pseudovirion. So just to make this clear to you, let's take a look at this virus that we have here, which has the outside envelope of B but the inner genome of A. You will realize that in the pseudovirion, it will contain the same genetic material and capsid as virus A. However, remember that it is the virus B envelope that determines the infectivity pattern of the pseudovirion. However, this fact is only true for the current generation. Does that make sense to you? I'll repeat this one more time. It is basically this outer envelope that will determine the infectivity pattern of the pseudovirion and not the inside genome and capsid.